Hello everyone. Today I'll be explaining an AP Bio related lesson, which is about prokaryotic cells. First, I'll be introducing myself. My name is Shahad Imad Al-Harbi. I'm a 12th grader from Dubai National School. Let's start by first knowing what is a cell, as a basic start for our lesson. A cell is the smallest structural and functional unit of an organism, which is typically microscopic and consists of cytoplasm and a nucleus enclosed in a membrane. A cell has two types, which are prokaryotic cells and eukaryotic cells. Each are present in certain creatures and have different structures and functions depending on where are they present. Today, I'll be discussing prokaryotic cells and focusing on the different aspects within the cell. Prokaryotic cells are placed in two taxonomic domains, which are bacteria and archaea. So some of the characteristics of bacteria is that it can cause diseases, though they are environmentally important and vital as decomposers, and they are useful in manufacturing products and drugs. On the other hand, archaea are capable of living in extreme habitat, such as in the bottom of the ocean, and even thrive in petroleum deposits deep underground. Prokaryotic have different shapes and sizes, including three basic shapes. First is coccus, which is any spherical or roughly spherical bacterium, and it is displayed as the purple spheres on the screen. Bacillus is the term used to describe the rod shape of a certain bacteria, which can also be attached to each other. The two types of spiral cells are spirillum and spirochete, with the spirillum being rigid with external flagella, such as displayed on the far right, and spirochete being flexible with internal flagella, such as in the left side. Prokaryotes' wall structure contains three main walls, which are plasma membrane, cell wall, and finally the glycocalyx. First, the plasma membrane, which is a lipid bilayer with embedded and peripheral proteins. Then lies the cell wall, which is helpful in maintaining the shape of the cell and is supported and strengthened by peptidoglycans. And then the glycocalyx, which is composed of layers of polysaccharides on the outside of the cell wall, and when the glycocalyx is well organized, rigid and resistant to any kind of removal, it is called a capsule. Leaving the outside structure, let's focus on the inner side of the prokaryotic cell, which contains the following structures. The cytoplasm is a thick solution that fills each cell and is enclosed by the cell membrane. It is mainly composed of water, salts, and proteins. The nucleoid, which means nucleus-like, is an irregular shaped region within the prokaryotic cells that contains all or most of the genetic material. The chromosome of a prokaryote is circular, and its length is very large compared to the cell dimensions, so it needs to be compacted in order to fit. A plasmid is a small, often circular DNA molecule found in bacteria and other cells. Plasmids are separate from the bacterial chromosomes and replicate independently, often. They generally carry only a small number of genes, some associated with antibiotic resistance. A ribosome is a cellular particle made of RNA and protein that serves as the site for protein synthesis in the cell. Flagella, hair-like structure that acts primarily as an organelle for locomotion in the cells of many living organisms. Fimbriae are long protein structures located at the surface of bacterial cells. They enable the bacteria to bind to specific receptor structures and thereby to colonize specific surfaces. Conjunction pili allow the transfer of DNA between bacteria in the process of bacterial conjunctions because they allow for the exchange of genes via the formation of pairs. What makes prokaryotic cells different? A prokaryotic cell is simpler and requires far fewer genes to function than the eukaryotic cell. Therefore, it contains only one circular DNA molecule and various small DNA called plasmids. Prokaryotes are unicellular organisms that lack membrane-bound structures. Fossil records indicate that eukaryotes evolved from prokaryotes somewhere between 1.5 to 2 billion years ago. Two proposed pathways describe the invasion of prokaryote cells by two smaller prokaryote cells. They subsequently became successfully included as part of a now much larger cell with additional structures and capable of additional functions, and it is referred to as the endosymbiotic theory. And here we reach the conclusion of this video. I would like to thank you for watching, and I hope all the points explained were clear.